Welcome to my YouTube channel. In last video I discussed about the photosynthesis pigments takes place in blue and red regions of the spectrum mainly. So uh, some photosynthesis does not takes place at the wavelengths of their visible spectrum. So chlorophyll is the major pigment responsible trapping light. Other thylakoid pigments like chlorophyll B, A, carotenoid, xanthophyll. So these are all also called as accessory pigments. So absorb light and transfer to chlorophyll A. So they not only enable a wider range of wavelength of incoming light to be utilized for photosynthesis, but also protect chlorophyll A from photooxidation. So next, coming to the light reaction. Light reaction is also known as the photochemical phase. So it includes the light absorption and water splitting, oxygen release and formation of high energy chemical intermediates like ATP and NADPH. So several protein complexes are involved in this process. It has two systems like photosystem 1 and photosystem 2, and light harvesting complex within occupy in the photosystem 1 and 2. So during the light reaction, the light harvesting complex are made up of hundreds of the pigment molecule bound to the protein. Each photosystem has all pigments forming light harvesting system known as the antennae. These pigments helps to make photosynthesis more efficient by absorbing different wavelengths of the light. The single chlorophyll A molecule forms the reaction center. In photosystem 1, the reaction center chlorophyll A has an absorption peak at 700 nanometers. It is called as the P700. While in the photosystem 2, absorption maxima at 680, so it is known as the P680. So these are all of the light reaction. Here we can see this the pigment molecules. This is photon in the presence of the light. It is the primary acceptor, and here it is the reaction center. And then electron transport system. So in photosystem 2, the reaction center chlorophyll A absorbs 680 nanometers wavelength of the red light, causing electrons to become excited and jump into orbit further from the atomic nucleus. These electrons are picked up by the electron acceptor, which passes them to electron transport consisting of the cytochrome. This movement of electron is a downhill in terms of the oxidation, reduction or redox potential scale, and they passed on to the pigments of the photosystem 1. Simultaneously, electrons in the reaction center of PS1 are also excited. They receive the red light of the wavelength 700 nanometers transferred to the another acceptor molecule. So electron transport system, here you can see first it enters into the light harvesting acceptor photosystem 2 and then the photosystem 1. So it appears like the Z scheme process. So also known as the Z scheme. Z scheme process. The electrons in the reaction center of the system are excited when they receive red light of the wavelength 700 nanometer and transfer to the electron acceptor molecule. Has a rate redox potential. These electrons are then moved downhill again. This time to molecule of the rich NADP plus. The addition of this electron reduced to NADP plus to NADH plus 1. Buy coffee and so transfer of the electron starting from PS2 uphill to the acceptor, down the accept transport chain to. So it looks like the Z scheme due to characteristic shape. This shape is formed when all carriers are placed in the sequence on the redox potential scale. And coming to the splitting of the water. Electrons that moved from the photosystem 2 must be replaced. This is achieved by the electrons available due to splitting of the water. Splitting of water associated with photosystem 2. Water is split into 2H plus O and electron. This creates the oxygen, one of the net products of the photosynthesis. The electrons needed to replace those removed from photosystem 1 are provided by photosystem 2. We need to emphasize here that the water splitting complex is associated with photosystem 2. So in next we'll discuss about the cyclic and non-cyclic photophosphorylation process.